Okay, this is Dr. Launen from Keene State College, and I want to show you an example of a human genome chromosome, chromosome 1 specifically, to give you a feeling for the kind of information that you can get from a fully annotated, finished genome. So here we are in the University of California, Santa Cruz, UCSC, Genome Browser Gateway, which has a lot of really fantastic genomic information in it, including a lot of information about humans and also other model organisms. So we're looking at the human assembly, and we're looking at the most recent one available in this browser. And we're going to take a look at chromosome 1. Bear with me for a minute. This is going to take just a few seconds to load. Now already in this, even though the screen hasn't fully loaded, I can get a little bit of information. I can see the total size. It's over 248 million base pairs. It's a very large chromosome. We have a fairly big genome. There's the whole thing, and there's all the different genetic information that exists for this particular chromosome. If we look at this map shown vertically here, the different shades of gray are from banding patterns that have been determined by using what's called cytology. So you take chromosomes and you stain them with different kinds of stain, and then you take microscopic images of them, and it will show different densities of the chromosome, which is the way that we started, one of the ways that we started mapping and studying different types of chromosomes. This area here is the centromere. The Q and the P arm represent the arms or the sides of the chromosome on either side of the centromere, and the red box is showing us that we're looking right now at the entire chromosome. So I'm going to try to zoom in on the area right around the chromosome, and I'm going to do that by highlighting that area. Going to give it permission to jump to this new position. And it's going to take a few minutes because it's working with a lot of information. Let's see what genes are around the centromere region of chromosome 1 of Homo sapiens. Just kind of taking a little bit of a look-see here. All right, so now we are looking at the chromosome, or the centromere region, and these are lists of different genes that have been found in this location. So let's take a look at this Notch2 gene right here. I'm going to drill down into that gene, just sort of selecting it at random. And this is human gene, and its gene name is called Notch2. And here's a page that gives me just a ton of information about this gene, like more information than I could really possibly use. So I can see quickly that I can read a little bit of information here about what this gene encodes and what it's similar to. I can see that there are homologs or matching genes in other organisms like Drosophila, the fruit fly. I can see quickly that there's a transcript that includes the untranslated regions that has been sequenced. I can see that there are 34 exons in this gene, so between each exon I would know that there's an intron region. I can also do things like move down and look at the location. This is a membrane protein, not a cytosolic or an excreted protein. I can see that this protein is expressed in humans in the brain, the heart, kidney, lung, skeletal muscle, and liver, and that it's ubiquitously expressed in the embryo, which means all the cells in an embryo would express it. I can see that there are different diseases associated with mutations at this gene, and if I clicked on these areas, I could learn more about those diseases. I can see how different chemicals interact with this gene or gene product. I can see that there's what's called RNA-seq expression data, so people have extracted the RNA from human cells, and they have determined which kinds of cells this particular protein, the protein encoded by this gene, is found in most abundantly. Like, take a look over here and see these bars here. These are skin cells that are not exposed to sun, or skin cells that are exposed to sun, and it looks like skin cells that are exposed to sun express a little bit more of this protein than those that are not. That star indicates significance, that that data is significant. Let's see what else. I can take a look at the actual protein structure, and if I loaded the correct software on, which is really easy to do, I could spin that around and I could see alpha helices and pleated sheets and turns, where there are turns in these proteins. I can see that there's this protein is well known in rats and in zebrafish. And these are all, things that I could look at, a lot more closely. Let's see. There's a gene review, so if I wanted an article on a disease associated with this gene, I could click here and then that would give me the citation information and access to the article in all likelihood. Let's go back up to the top. Take a quick peek. Let's see. So up here we can see things like the Entree gene will usually give us information on the gene sequence. So here's the Notch2 protein. I'm still in the right area. This is showing me it's on chromosome 1. It's showing me the length of it. If I wanted to get the actual sequence, that's the assembly sequence, but if I wanted the actual sequence of the gene, I could drill in here. It would take me a couple levels of drilling, but let's see. This is showing me that it's in mice and in rats. Entree gene numbers here. That's going to probably take me back to the page I was just looking at. Anyway, let's see if we can find the actual gene sequence. I think we need to go back a couple of stages. It can take a little bit of time. Nucleotide sequences, which are in GenBank, and there's usually a few ways to get there, but there's the actual sequence as amino acid sequences. It's not A's, T's, C's, and G's. It's all the letters of the alphabet. So that's the actual sequence in amino acid sequence, the sequence of the actual peptide. And here's the actual DNA nucleotide genomic sequence. And I could easily download that file as well, just looking in here and saving it to file. So that's all I'm going to show you. I just want you to be aware that this is just like a snippet of the information that you can get from an annotated genome. So it allows you to really thoroughly study and investigate a sequence once it's been identified on a genome. You can identify the neighbors and so forth. And for the human genome, that's been really well established.